honored to be part of this wonderful event today. And I will be talking about extracellular stimulation using photovoltaic devices. So this is basically more or less an application, as you mentioned before, in bioelectronics. Let me first introduce our research group. So we are the In Vitro research group from Graz University of Technology. And this word in vitro is basically a mixture of two things. So we are working with in vitro experiments, doing cell experiments, and we are also trying to model and simulate what we are measuring and what we wanted to achieve. We are working a lot with extracellular stimulation and electrophysiology. What are the cell types that we are using? They are the most common ones: neuronal cells that we can find inside the brain, and cardiac muscle cells. These cell types are known to be stimulatable. They can fire action potential. So these are the excitable cells and most commonly used in electrophysiology. But we are also using non-excitable cells for electrophysiology experiments, as for example, lung cancer cells. So we are working a lot also in cancer electrophysiology. So let us first recap some basics of cellular function for those of you that are not really familiar with this topic. If we look at this generic animal cell here, that we can see that we have an extracellular space which is outside of the cell, and then we have all the things that the cell needs inside, but we are actually interested in the cell membrane. So if we are looking a little closer at the cell membrane, then, thank you, we can see that the cell membrane is actually a phospholipid bilayer. So this cell membrane helps to form a barrier between the environment and the cell. But of course, the cell has to exchange a few things also with the environment. And this is actually the target of the electrophysiology. We have some integral proteins that span the whole bilayer, and they allow, for example, the passage of different types of ions. Most of these channels are selective, so they do not let through any ion, but for example, they focus on potassium or chloride or sodium channels. From a modeling point of view, actually, the channel can be regarded as a resistor or the inverse as a conductivity, so it is modeled as a conductivity. Usually, these ion channels, they are not open all the time, so the ions cannot go through all the time. But these channels have the ability to change their state and switch in an inactive state or a closed state. This change in conformation of the protein is usually triggered by some gating mechanism. In this work, we will focus on voltage-gated ion channels. This means that actually the cell membrane acts like a capacitance, so it can be charged to a specific voltage, so we have a voltage drop between the inside and the outside. And this voltage difference between the inside and the outside of the cell leads to the gating of the ion channel. So in response to a change of the voltage, we might see that the ion channel is more likely to be open. This is reflected actually in the name of the ion channel with the superscript V for voltage. So this was just the basic introduction, and now we're looking at what we are actually wanting to do. We wanted to find a tool for neuronal stimulation with a high spatial and temporal resolution, and it should be little invasive and work without genetic modification, because, for example, in optogenetics, it is quite common to use light to manipulate the brain, but there you need a genetic modification, and we wanted to avoid this. So we came up with the idea of using a solar cell kind of device and combine it, with the neurons and see if we can make them fire an action potential. So we were not sure about if this would be working. Let's have a look at the working principle. So these are basically the devices that we are using. These are organic electrolytic photocapacitors, or in short, we call them OEPC devices for light-triggered external stimulation. And then we're looking here, we can see on the bottom the OEPC device, which consists of several layers. I will come to this later on. And on top, we see again this bilayer of the cellular membrane with one of these ion channel proteins. So what we do is we switch the light on. We hope that we can then depolarize the membrane of the cell, open these voltage-gated ion channels that I was talking about, and in the end, hopefully be able to fire and action potential of the cell. So what are the materials that we are using? We are probably more familiar with these materials. So we usually use a back electrode of ITO, and then we use vapor deposition to deposit 30 nanometer layers of an N-type and a P-type organic semiconductor. Why did we choose these materials? Because actually they are rather cheap, so they can be easily used for our experiments. They are non-toxic for the cells. They are easy to use, as you mentioned before. It's easy to fabricate them. We can shape the size of the cell, of the solar cell, and also the shape. And in addition, we can adjust the capacitance of the whole thing using, for example, P.PSS code. So the first question that we have, do the cells actually like the material? Will they survive? So we performed some cytotoxicity tests. Here we see an enzyme which is called ADH, which is usually inside the cell. So if we find it in the medium outside, then this gives a hint that actually there must be some leakage, or there must be some damage to the cell. And if you're now looking at the results, we compare this with glass, because this is where we usually grow the cells. And we can see that actually our OEPC devices perform similar to glass. So after 3 weeks, we see that there's actually no difference to glass, so the cells seem to be happy on these materials. The next question that we wanted to answer was, what is actually the electric field that the cells can feel? So we can see here how we measure this. We have the electrolytic bath, where the cell lives in. We have the OEPC device, and then we can measure the electric field that arises from switching the light on. We then were trying to model the behavior of the cell to get an idea what happens when we switch the light on. In this first step, we were using a photodiode equivalent circuit that was parameterized based on our measurements. And actually, before we started to plant the neurons on our devices, we started with some model cell, which is very often used in electrophysiology. It's the HEC 293 cell line. And the good thing about it is that we can actually let the cell express any ion channel that we want. So in this case, we chose a TEC RFP KB1.3. So if we're looking at the name of this channel now, we can see the K for potassium and the V for voltage gated. So we have a potassium channel which is voltage gated and fits our needs. Why is this channel so nice for our ideas? Because when I change the voltage of the cell, which I can do experimentally, this channel immediately opens, so the kinetics are really fast. And when I leave the voltage the same, then the channel doesn't close. So we thought this might be a good idea to get a first impression of what actually the OEPC devices are doing to these voltage-gated ion channels. So the question that we wanted to answer, can we now stimulate these voltage-gated ion channels and increase the conductivity of our cell? For this, planted our HEC cells on the OEPC devices, stimulated them, and performed patch clamp experiments. With the patch clamp experiments, we can actually force the cell membrane to a specific membrane potential, and at the same time measure the current through the cell. These experiments are then used and fed into our simulation models. This seems easy, but actually we need to consider quite a few things. We need a model for the cell, the kinetics of the ion channel need to be considered. We need also to model the patch clamp amplifier, which is very often left out, but you need to keep in mind that the amplifier is trying to keep the voltage as a specific voltage level, and you perform external stimulation. This is a huge disturbance for the amplifier, and the amplifier will try to work against it. So we need to include that in the model. And of course, the electrical response to light stimulus for the OEPC device, and also the interaction between the OEPC device and the electrolyte on top, because this is something that solar cells usually don't have. As we heard before, we're trying actually to protect them against moisture, and we soak them in an electrolytic bath, so this is a completely different story. So how does this actually work when we're looking at the cell model? We started with this very simple channel with a simple kinetic, but each of these ion channels can be in different states. They can be open, they can be closed, or inactive. The thing is, there are not only two different states like open and closed, but there are many, many different states. But we can only see when the channel is open. We do not see if it's inactive or closed when we're just measuring the current, because when there's no current flowing, we just do know it's not open, but we don't know what else is going on. So usually these ion channels can be modeled by so-called hidden Markov models. These are models where you can define different states and they are hidden, so you don't see in which state the channel actually is. So this is quite a nice representation of an ion channel. We can see, for example, here a representation of a very simple kinetic with 5 different closed states, an open state and 1 inactive state. And in the open state, we see the current flowing. So if we implemented the states for our specific channel, it's actually a very simple structure. We can then compute the open probability of each of these specific ion channels individually. These open probabilities depend on the voltage that the cell sees and also on time. And with this open probability for each individual pore, we can just sum up over all the pores to get the macroscopic currents, to get the whole current that is flowing through the cell, and that we then see in the patch clamp measurements. So we end up with a voltage-dependent model for the conductivity of our ion channel. So here we have some results. We applied different protocols to the cell, so we were starting to clamp the cell at about minus 100 millivolt. This means in this case all these potassium channels are closed, so there's no current flowing, apart from some leakage. And then we start clamping it to higher voltage levels, and we see that at about minus 40 millivolt, the ion channel starts opening up, and this is what you see here with these traces on the side. With increasing voltage, the current increases too. And in red, you see the simulations and the black T measurements. So this is what the entire model then looks like. So we have the OEPC model, we have a FEM simulation which is trying to model the interface between the electrolyte and the OEPC. We have a very sophisticated cell model and also the patch clamp amplifier with all the compensation that the amplifier is applying. These are now the first results of our hex cells, so of the very simple cells. We again applied these voltage steps to the cellular membrane, and while the cell was clamped, we applied light pulse, and we can see that on the one hand the current increases, which is voltage step, and here you also see the 5 millisecond light. We can also see that the current increases a lot during this 5 millisecond period. And again, we compared the measurement to the simulation and got a quite good agreement. So what does this mean to the conductivity of our ion channel? We see here that in black, this is without light, so just clamping the cell to different membrane levels. We can see that we reach half of the maximum conductivity at about minus 15 millivolts, and in red, we see the maximum light intensity. We can see that here, we already achieve half of the maximum conductivity at about minus 40 millivolts. So we were really able to put more voltage across the membrane of the cell and increase the current flowing through the cell. And now I have a video where you can actually see what the cell is doing. So we start clamping the cell to about minus 100 millivolts, so it's dark blue. Then we clamp it to about minus 40 millivolt, and we can see that it's getting lighter blue, so the cell is now clamped to minus 40 millivolt. And then we will soon see that the light is actually switched on, and we get a huge reaction of the cell. So you can see that the cell membrane changes the color, and it's reaching up to about 80 millivolt for a rather short period of time, but this is enough to open these ion channels. So I'm now trying to answer the question, can we stimulate these voltage-gated ion channels using an LED? And the answer is yes, of course. So at minus 100 millivolt, all the ion channels are closed. At minus 40 millivolt, we can already see that the ion channels start opening, so we get higher conductivity of the ion channel, and if we switch light on, we can again push the conductivity of the channel, in this case of about 40%. So yes, we can stimulate. But as I said in the beginning, we're actually more interested in the question, can we actually make the neurons fire? So we have a really nice picture here of some neurons which are grown on our OEPC devices, and they are happy there for about 2 to 3 weeks, so they seem to like the material. And the answer is, it seems that we can achieve an action potential, so we compared the results to a current injection. So again, we put a patch clamp needle inside the cell, we injected the current to help the cell to fire, and then we switched its current off and used a 3 millisecond laser pulse to make the cells fire. And we can see that sometimes it fails. So when you see the red light on, there's no action potential, but most of the time we can actually achieve the cell firing and action potential. So, to answer the question, it seems to work. We can use these optoelectronic devices and make the neurons fire. The next step we did was to use these devices as neuronal implants. This is still work in progress. So these devices are currently implanted into an animal model and stimulated for about half an hour. And then we're looking at the C-force expression. The C-force expression can be a measure for the activity of the neuronal cells. And we see here on in the middle the stimulation group, where you see these, hopefully these light dot, and this means that there is a higher activity of the neurons. So these devices are working also inside the animal model as an implant. So then we came up with the question, what about different cell types? Can they also be stimulated? But for example, about cardiac myocytes, and we were there looking at a specific, very well-known channel, which is the L-type calcium channel, or ICAL. In this case, we have a rather sophisticated cell model. So you can see here there's not only one single ion channel, as I showed you before for the HEC cells, but actually there are quite a few ion channels, pumps, and exchangers. And we also used a multi-compartment model to take into account different local calcium concentrations. Because we were focusing on this ICAL ion channel, and if we're having a closer look at this ICAL ion channel, then we can actually see that this is now the Markov state diagram, which in comparison is more complex than the one that we saw before, because this ion channel not only depends on the voltage, but also on the local calcium concentration. So inside the cell, these channels are co-located with calcium channels, and so the current through this ion channel can be regulated by voltage and by calcium concentration. And if we're looking at the results, we can see that here, in response to 100 millisecond light pulse, we can see that actually our simulation fits nicely to what we could measure. And just to give you an idea how much the light intensity may influence the current that we see, we performed measurements with different light intensities that you can see here, and we can see that actually the current that we can see changes a lot with the light intensity, which for us means that we also can change the voltage that the cellular membrane can see. So this brings me to my last question. When does the stimulation actually fail? I would say very often, because there are so many things that you need to consider. There's one thing which is rather well known. When the cell is too far away from the device, what happens? This is actually what you see on the slide here. All the current will just bypass the cell and not go into the cell, but just flow underneath the cell somewhere else. And the cellular membrane will not see any change in voltage, and nothing is going to happen. This is actually something that happens with cells that do not attach nicely when you perform in vitro single cell experiments. What is also important is the cell site relative to the electrode size. So they both depend on each other. The type of the cell, so how big is the cell, what ion channels can be there, how big is the resistance of the membrane, and the current density that we can achieve through the stimulating device. There are many more parameters, and this is still ongoing research, what is actually the best device that you can use. So this brings me now to my conclusion. We have implemented these OEPC devices that are able to activate voltage-gated ion channels and induce neuronal action potentials. So we have now implemented an optically controlled in vitro experimental tool, and we're trying to lay a mechanistic understanding for the light-triggered extracellular stimulation. So to conclude, we can say that they are actually a really nice experimental tool for our research. So with this, I want to thank our scientific collaborators, especially from the Central European Institute of Technology in Brno and from the University of Zagreb. And this whole project was funded by the Austrian Science Funds. So thank you very much, and I'm now very happy to answer your questions.